Okay, we ended part 1 of chapter 12 lecture video by talking about Wallerian degeneration and how whether that degeneration happens, or that sort of recovery of the axon happens on its own, is up to the cell itself. But what about medically speaking? How can we intervene for helping an axon to regrow or repair of a neuron? Are we able to do that? Currently, where are we at in terms of research, in terms of, having new innovative way of repairing these neurons? So this video kind of goes over some of the advances we're making in assisting with neuron repair. So we'll go through this video first before we dive into the formation of an action potential.
Combat, cancer, and accidents all can cause devastating nerve injuries. Sometimes the body heals on its own.
Your peripheral nerves, so the ones in the arms and the face, have an inherent ability to regenerate, but only under ideal circumstances.
With support from the National Science Foundation, University of Florida biomedical engineer Christine Schmidt is working to restore nerve function when injuries are more complicated.
You took that muscle and rotated it, took it over the back of his elbow.
Surgeons can sometimes move a nerve from one part of a patient's body to another. Schmidt has developed a method that grafts cadaver tissue onto the damaged area to act as a scaffold for nerves to regrow themselves.
Basically what we're doing is removing all the cellular material that would cause rejection, but leave behind the native architecture. So you're putting this graft into the site of injury. And now that graft is providing a scaffold for blood vessels to grow in, and then once you have that recellularization, your nerve fibers can then regrow and then ultimately regain that muscle function.
Navy veteran Edward Bonfiglio, wounded in Afghanistan, faced the prospect of an amputation. A graft was a welcome option. The company Axogen distributes the grafts, which were developed based on work done in Schmidt's lab.
And his family pressed the doctors to say, are there any alternatives? He was a young, healthy, vibrant guy. And they had a great surgeon at Walter Reed who was willing to work with them to find those options. Is this some of the micronized nerve that you're working with?
Schmidt and her team are also looking at other approaches to directly stimulate nerve growth, using natural sugar molecules found in the body as building blocks, eliminating the need to transplant tissue.
So you don't have to actually take it from somebody's body, you can grow it.
While the ultimate goal in nerve regeneration is reversing paralysis, Schmidt says intermediate successes, like improving lung or bladder function, can be invaluable to patients and their families.
So rather than saying we're going to try to tackle this humongously complex beast and try to get the patient to necessarily be exactly like they were before, why not provide some function that will have merit to this patient?
So after a serious injury, it's not just about getting your nerve back, it's growing it back. For Science Nation, I'm Miles O'Brien.
Okay, so that was interesting. And now we're going to dive into the more difficult topic of this chapter, which is about how an action potential is generated within a neuron, biochemically speaking. So we'll begin by talking about the ion content that is outside and inside of the neuron. The electrochemical gradient is what drives the movement of ions. We talked about, gradient, chemical gradient versus an electrogradient in general biology class. Now it is bringing them back and applying them in terms of a neuron. Now, this gradient is actually a sum of both chemical and electrical forces acting on the specific ion across the plasma membrane, whether it be sodium and potassium. Okay, these are the two critical ions we'll talk about for action potential to be propagated. Okay, so that's why we'll zoom in and talk about these two main ions. Now, a chemical gradient, just as a reminder, is dependent on concentrations of ions. We talked about diffusion. Things always move from a higher concentration to a lower concentration. So that's according to its chemical gradient. The other thing that drives movement of ions, because ions are charged, they're either positively charged or negatively charged. So these can be driven by what we call electrical current as well, because you have attractive forces involved. Under resting membrane potential, before arrival of an action potential, the inside of the neuron, or any cell for that matter, is more negatively charged than outside of the cell. And that's due to two things: the presence of negatively charged proteins like this, and the fact that, it is actually easier for potassium to diffuse out of the cell through what we call leaky potassium channels. Like this. We won't talk about leaky potassium channels much, but, just want you to understand why the interior of the cytoplasm tends to be more negative. But more positive charges moving outwards than coming in, and the presence of these negatively charged proteins is what result in excess of negative charges inside the cell when compared to the outside of the cell. So that is why the resting membrane potential is a negative number usually. And for a neuron, it usually averages to be about negative 70 millivolts. So because you have that negative charge interior, so that's why you have this electrical current, because positive charge ions will be more attracted towards the inside of the cell than outside of the cell, electrically speaking. Okay, so now let's look at the two ions of interest, which is potassium and sodium. We'll compare whether they tend to move out of the cell or move into the cell. So for potassium, the intracellular concentration of potassium ions is a lot higher on the inside of the cell when compared to the outside. And hence, according to potassium ions' chemical gradient, it wants to drive the potassium out of the cell. Now, an electrical current also exists for potassium ions. And we talked about how a positive charge ion would want to be attracted towards the inside. So that's why it actually opposes the chemical gradient, because the positively charged potassium ion is attracted to the negative charged cytosol on the inside of the cell. But the chemical gradient, thankfully, actually wins slightly and overpower the electrical gradient. Okay, and because it is able to overpower the electrical gradient, that's what drives potassium ions out of the cell versus coming in. The magnitude of force is greatly weakened by the electrical gradient, but still, potassium ions tend to move from inside of the cell towards the outside of the cell if there was a channel available. Okay, so this is what acts as a driving force for potassium ions to exit the cell during an action potential. And we'll talk about that in a bit. Now, so that's for potassium ion. Now we talk about sodium ions. Which way do they go? So for sodium, the extracellular concentration of sodium ions is a lot higher than the inside of the cell. So that's why you see there's a lot more purple balls represented as sodium ions than the inside. And hence, according to chemical gradient, it drives sodium into the cell, going from a higher concentration to an area of lower concentration. So this is the chemical gradient that drives sodium ions into the cell. And in addition, sodium ions is being is, itself being positively charged, is attracted to the negatively charged cytosol. So electrical gradient further enhances the movement of sodium ions from the outside towards the inside the cell, and hence the net sodium electrochemical gradient that drives sodium into the cell is very, very large, is huge, okay? Double the force. So this is why sodium ions rushes into the neuron very quickly as soon as those sodium ion gates open upon the arrival of an action potential that we'll talk about. And hence, what's going on here is that these electrochemical gradient is providing a source of potential energy that is stored like, for example, water behind a dam. And the cell membrane is acting like a dam that is stopping the flow of these charges until the right timing comes, which is arrival of a stimulus. The cell that tells the neuron, hey, you got to get this going so we can get that signal onto the next neuron. Now, without the dam, which is the cellular membrane, the ions, technically speaking, will just diffuse freely to equilibrium. But because there's a cell membrane right here that sits between extracellular space versus intracellular cytosol, that's why we can have different ion concentration on either side of the membrane and reset it back that way for the next action potential to come over, okay? So in addition, under resting membrane potential, okay, note that we're talking about resting membrane potential, there is a very critical pump that sets the ions on the two sides of the cell membrane. And this is called the sodium-potassium exchange pump, which I've slightly glossed over in the cell chapter previously. And I talk about this exchange pump immensely in 1406 as well when I teach general biology. So this pump will eject 3 sodium ions for every 2 potassium ions that come into the cell. So because once after, these ions are switched spots after action potential has, undergone its route, okay, there needs to be something that reset the ions back to their original position. So that's the job of the sodium-potassium pump, is to reset those ions back to their original position. And also, this is why during resting membrane potential, the outside of the neuron would be filled with sodium ion and inside of the neuron is filled with potassium ion. And the other thing is why this also contributes, slightly, so why interior of the cell is more negative compared to the outside, because you're pumping out more positive charges out of the cell compared to positive charges that you're bringing into the cell. So there are two things that the sodium-potassium pump is serving for, okay? To reset the ions in their original position, and second is to contribute somewhat to that negative overall membrane potential in a resting neuron. Okay. And before I go into depth of each step of an action potential, we need to discuss a few more things. There are 2 different types of active ion channels in neurons. One is called chemical gated ion channel, which is when activated or open, when we call something activated, a channel activated, it means that it's opening. And when it opens, it opens because it binds to a specific chemical, for example, like a neurotransmitter. We talked about acetylcholine in the neuromuscular junction in the previous chapter. So acetylcholine is that chemical that binds to that specific site on this chemical gated ion channel, specifically sodium ion channel. And upon binding to that chemical, that's when the gate opens, channel opens, allows sodium ions to flow inwards into the cell. So this is why these are called chemically gated ion channels, because they are activated by binding of a chemical. Okay, and because neurotransmitter is usually released into the synaptic cleft, remember we talked about that neuromuscular junction, and it's for binding to the next neuron, or in the neuromuscular junction's case, binding to the muscle fiber itself. That's why these chemical gated ion channels tend to be abundant on the dendrites and cell body versus the axon or the axon terminal, because these are usually for receiving a stimulus coming through, okay? Because it needs to wait for the arrival of neurotransmitter. Remember, we talked about dendrites, cell body, therefore receiving signal, okay? So that's why chemical gated ion channels tend to be on the cell body or the dendrite side versus the axon. And hence, the other type of ion channel is called the voltage gated channel, which tends to be on the axon side, okay? So these gated channels has a gate that will open and close in response to the cell's membrane potential, okay? So these are electrically activated, okay? These are mainly for propagation of an action potential, so they're abundant along the axons of either unipolar or multipolar neuron, depending whether you're talking about those neurons in the peripheral nervous system or the central nervous system. So for example, sodium channels, voltage gated sodium ion channels, have two gates, okay? It actually activate upon the activation gate will open upon stimulation, which for the sodium ions case of a neuron is anywhere from -60 to -55 millivolts. Okay, that's how we measure membrane potential, is by voltage, because it's electrical differences. It's kind of like a battery, okay? And when it reaches -60 or -55 from -70, that's what activates the opening of these activation gate. When the potential reaches a positive value to +30 millivolts, it causes the inactivation gate to close, and that stops the sodium ion entry. So here's that activation gate, and here's that inactivation gate. Activation gate opens when it is at -60, and for the simplicity, we're just going to refer it to -60, so we don't have to say -60 to -55 every single time, but that's the range value, and your book went with the -60 in their illustration, so that's why we're going to just talk about it in sense of -60 millivolts. And when the membrane potential gets up to +30, that's when the inactivation gate will close, stopping the flow of sodium ions. So that's why we call these voltage gated channels, because it's driven by what the membrane potential is, whether it stays open or closed. And these are called chemically gated ion channels because it requires a chemical to bind at the site in order for it to open or close. All right, so remember that the membrane potential is a measurement, or defined as the measurement of the cell's polarization situation. And at rest, before arrival of a stimulus, the membrane potential should be overall negative, with what I talked about earlier, those presence of negative charge proteins and also the imbalance of positive charge movement by your sodium potassium pump. So a neuron's resting membrane potential is about -70 millivolts, while that of a muscle fiber is -85 millivolts. So we're mainly going to talk about neurons for this chapter because it's a chapter on nervous tissue, but they're slightly different between neuron versus muscle fiber when we talk about these voltages. Now, keep in mind that the cell membrane is, like we talked about, acting like a dam that keeps the ions separated until those gates we just discussed opens, kind of like punching holes in the dam to allow the water to come through. And in this case, the water are the ions, and they're just waiting around until somewhere opens and they rush through. So when a stimulus does arrive at the neuron and opens those specific ion channels, for example, when the membrane potential gets positive enough to open the voltage-gated sodium channels, which we already talked about negative 60 to negative 55 millivolts, the sodium ions will then rush in to the neuron in a process called depolarization. The membrane potential will then become very positive due to that influx of positively charged sodium ions all over inside the cytoplasm of the neuron. Okay, and it gets to the point where then it closes the voltage-gated sodium channel, and then it opens the voltage-gated potassium channels instead and allow potassium ions to flow outwards so that your potassium ions are now abundant in the extracellular space of the neuron. So this ejection of potassium ions or moving out of the cell will set the membrane potential back down to negative 70 millivolts because you're ejecting positively charged stuff outside of the cell. This process is called repolarization because you're bringing back the membrane potential back to its resting units, okay, back to its resting membrane potential, which is negative 70 millivolts, okay? So at first it was negative 70 millivolts. Then once it reaches negative 60, for example, sodium channels open, and then you have influx of sodium ions until it reaches positive 30 millivolts, and then your potassium ions channels open and allow potassium channels to open. Sodium ion channels are now closed, so no more sodium ion flowing through anymore, and the membrane repolarizes back to negative 70 millivolts. So that's the sort of a nutshell of what's happening in an action potential. It involves depolarization and repolarization, and it actually has a third thing that happens called hyperpolarization, which I'll talk about when we get to the different steps of an action potential. Excuse me. Okay. So all these different direction of ion movement that cause depolarization and repolarization to happen is what produces an electrical impulse, or what we call an action potential, to be propagated along the entire cell's membrane, and in the neuron's case, the entire axon lemma. So here's your neuron, and then the axon, and then the axon terminal. So it's allowing the action potential to move this way down the entire length of the axon during the action potential. Okay, so opening of sodium ion channels will result in depolarization that will increase the membrane potential from negative 70 millivolts to positive 30 millivolts, and opening potassium channels will lead to repolarization that will decrease the membrane potential back down to negative 70 millivolts. Now, technically speaking, when the membrane potential is increasing due to activation of sodium ion channels, a second stimulus cannot arrive because the sodium channels are already open. So this is a little bit different from muscle fiber when we talk about summation, wave summation of a TREP, for example. Okay, how we talked about a twitch and how they have to be added together, and you don't have to allow the muscle fiber to relax entirely. But for nervous system is a different story, because we're talking about these ion channels opening. And once ion channels, or sodium ion channels specifically, are open, you cannot ask them to open any further. They're already open. So We call this, shutdown of the second action potential arriving. This is called the refractory period. Okay, refractory period has to do with further depolarization cannot occur until the whole thing is done, because the sodium channels are already open. You can't make it open any more than it is already doing, okay? And hence, further depolarization cannot happen until the whole action potential is done. Okay, so the other thing that needs to happen to reset the ions to the correct concentration on either side of the membrane is the ion reset of position. I'm going to explain this one more time. So remember that a membrane at resting potential has sodium on the outside. You see lots of sodium ions and then lots of potassium ions on the inside. After an action potential has gone through, remember all those sodium ions on the outside are going to now influx into the interior part of the cell, and then all the potassium that are on the inside will now move outwards towards the outside of the cell. So after an action potential has happened, you have buildup of potassium ions on the outside and buildup of sodium ions on the inside. So in order for an action potential to continuously keep going, the ions eventually have to be reset back to their original position with more sodium ions on the outside and more potassium ions in the inside, because this, technically speaking, is the reset of the neuron. Okay, reset of neuron, where we have negative 70 millivolts as the membrane potential, and the sodium ions are lots of them on the outside and lots of potassium ion on the inside. Okay, so that's why the sodium potassium pump, which will pump out 3 sodium ions for every 2 potassium that goes in the cell, will maintain this differences in sodium ion concentration on the outside versus potassium ion concentration on the inside. Okay, so there would be another membrane here, transmembrane protein, that acts as your sodium potassium channel that reverts these ions back to their original position. Okay, so again, the other reason for why we have to have the action of sodium potassium pump is that it further contributes to the overall negative membrane potential as well. Now, if we talk about, in the sense of one action potential, it actually doesn't require the potassium pump to be activated after every single action potential. There's just lots of sodium ions out in the extracellular space and lots of potassium ions in the cell. So one action potential is not going to kill that, concentration difference. Sometimes thousands of action potential can go through before, it offsets the ion concentration. But if you think about it in a maximally stimulated neuron, okay? So, the job, like I said, the job of the exchange pump is not essential to any single action potential. But when a neuron is maximally stimulated, it can generate thousands of action potential per second. Okay, that's how fast a neuron has to work when it is maximally stimulated. And hence, eventually, the role of sodium potassium pump become a critical factor in regeneration of an action potential, because if the ions are not in their correct side of the membrane, a neuron cannot conduct the electrical impulse and hence loses its function. Okay, so it's always gotta be more sodium ions on the outside and more potassium ion on the inside for an action potential to happen. If the neuron somehow stays this way, for example, sodium potassium pump requires ATP to do its job. It's an active transport, so that's why it requires ATP, and that's why we call it a pump. pump because it actually needs energy input into the pump in order to do its job. So if somehow the neuron does not have enough ATP supply, or something went wrong with ATP synthase, that is not generating enough ATP, the neuron, technically speaking, can shut down and no longer does what it's supposed to do, because they need that constant supply of ATP. One of the reason is to power the sodium potassium pump, so that it can always set those ions back to their original position. So that's what we call it. It regulates and maintains that ion differences between the outside of the neuron and inside of the neuron. Okay? All right. So neurons require a lot of ATP to power. Okay? Especially when there are cases when we need to really stimulate the neurons for a prolonged period of time. All right, now before I go into what a graded potential is, let's look at what happens within a neuron in an overview. There's a figure in my 2004 A&P textbook that, is actually a 6th edition of your textbook now, is when I took A&P in college. But they had this great overview illustration that was removed from the newer version, and I want to draw this for you. So what happens is you have this neuron, okay, the cell body, which then can narrow down to the axon. And then you have your telodendria that can end in axon terminal, for example. Okay, so this cell body part of it with dendrites branching out, okay, for example. So all of this, originally speaking, it's the membrane here within the cell body is at first at resting membrane potential. Now, what needs to happen is, let's say this is synapsing on another one axon terminal from another neuron. Okay, this is neuron number 1. This is neuron number 2. And then this also then synapse on another neuron with its dendrites, okay? So this is neuron number 3, okay? So neuron 1 sends a signal, let's say release of a neurotransmitter, okay? And that will activate those sodium ion channels within this dendrite of neuron number 2. Influx of sodium ions becomes possible because now you activated those chemically gated sodium ion channel. Now, just because 1 or 2 sodium ions come through doesn't mean that it's going to be enough to cause an action potential to trigger down the axon. Okay, enough amount of those stimulus needs to come through and allow more sodium ions to arrive, and they kind of collect at this site called the axon hillock. Okay, so we call these graded potential. Okay, so for example, this stimulus, a stimulus can cause your membrane potential to go from negative 70 millivolts to, let's say, negative 65 millivolts. Well, that's not enough to open those voltage-gated sodium channels at the axon, okay? So this then goes back down to negative 70 maybe if this stimulus doesn't continue to give off that same signal, okay? So enough amount of these graded potential has to be generated to get to a point so that your membrane potential within the cell body at the axon hillock here gets to be about negative 60 to negative 55 millivolts. That's what's going to trigger an action potential to start potential. Okay, to start down this way of the axon. Okay, so let me make these different colors. So this is that resting potential in the beginning, okay, which then leads to graded potential as a second step when this neuron number 1 releases those neurotransmitter to try to activate your neuron number 2. Once graded potential generate enough of a difference of membrane potential, then it's going to activate an action potential down the axon. Okay, and once the action potential then travels to the axon terminal, that's when synaptic activity can occur. Okay, so the last step of this whole story is the synaptic, I can't write over there, synaptic activity. Okay, synaptic activity, which is either a synapses with a neuron number 3 here, or if this is a neuromuscular junction, this can also, technically speaking, synapse on a muscle fiber. Okay, so that can be a motor end plate right there where that axon terminal nub is sitting on, and it synapse on the neuromuscular junction of the muscle fiber and release acetylcholine there due to being activated by this neuron 1. So that's what wraps up this whole story. Is we were talking about this part in chapter 10, how axon terminal is able to start a muscle contraction in the muscle fiber. But what happens before that is this whole story with the nervous tissue in this chapter 12. Okay, it all starts with a resting membrane potential that leads to graded potential because of a stimulus arrival. For example, pain, temperature, whatever it is that's coming through as sensory information that activated something that releases these neurotransmitter. Then enough of that graded potential is then going to activate that action potential that's going to relay that whole electrical signal to the axon terminal to tell your muscle fiber, for example, to contract and remove your hand from the source of pain, for example. Okay, so this is what sort of wraps up that whole story, how your electrical wiring of the nervous system is able to activate a response in the effector tissue. Okay? All right. So that's what that whole graded potential is about. And now we can talk about what is graded potential. So hence, before an action potential can happen, you need to accumulate enough amount of graded potential to take place in order to activate an action potential. For example, let's say a neurotransmitter, like I was talking about, arrives at that chemical gated sodium channel, opens it. Okay, so here's that, let me change back to red. Okay, here's that acetylcholine, for example. Okay, and now it arrives and stick in that binding site of the sodium ion channel, activates it. Now sodium ion rushes through and increases the membrane to slightly more positive. But for example, like what I was talking about, if that slight change in membrane potential is not enough to trigger activation of a voltage gated sodium channel at the axon hillock, okay, so it needs to get to at least -60 for that to happen. No action potential can be triggered just by a -65 millivolt change. Okay, so that's why these, in this particular scenario right here, it's depicting a graded potential, and it may take multiple graded potential in order for the membrane to get positive enough. And in the case of our voltage gated sodium channel, like I said, this has to be about -60 to -55 millivolts. So make sure you actually have to remember these numbers for the exam. Now, to keep things simple, we will just use, like I said, the -60, so I don't have to keep saying -60 to -55. What I mean when I say -60 millivolts is that range between -60 and -55, and that's what the textbook did here too. in their figures. And once the membrane potential is reached, then it's kind of like pulling a trigger on a gun, really. It fires an action potential down the entire length of the axon in one direction. And once it's fired, there's no turning back. You can't rest an axon that is undergoing an action potential. It will continue to fire until it reaches the axon terminal, followed by synaptic activity that I talked about to release neurotransmitter into the synaptic cleft that either synapse on a neuron, another neuron, or at a muscle fiber in the case of an NMJ. So any stimulus that opens a gated channel is what we call a graded deep potential. And for example, during a stimulation, okay, which is what I was talking about earlier, is that acetylcholine binding causing depolarization to happen. Now, the other thing about graded potential is that it can lead to spread of sodium along the inner surface of the neuron right here, and we call that generating a local current. So this part of graded potential explains why once an action potential is fired at the axon hillock, it leads to a chain reaction that opens the rest of the sodium channels down the axon. There's many sodium channels down the length of an axon, especially if we're talking about a myelinated axon. And the flow of sodium ion is kind of like a little wave, human wave, okay? So here comes sodium ions flooding through. It causes this part of the membrane to be more positive, which then relays and starts this gated channel, which then now allows sodium ions to move in here, goes to the next section to move in here, goes to the next section to move in there. So that whole local generation of this local current inside the neuron is what allows a chain reaction to happen and allows signal to be propagated down the membrane. Okay, it activated one channel after another. Okay, all right. So, and when, of course, when the stimulus is removed, we close the channels up, sodium ions are no longer moving inwards, and repolarization needs to happen to reset the neuron back to its resting membrane potential. All right. So, but if the chemical stimulus leads to a graded potential that is enough to activate the voltage-gated sodium ion channels at the axon hillock, enough meaning reaching about -60 millivolts, then depolarization of the initial segment of the axon becomes an all or none principle, which is what I was talking about. Once you fire the trigger, it goes on. It doesn't stop, okay? And it will reach all the way down to the axon terminal at the end of the neuron. And this activation membrane potential of -60 millivolts is known as the threshold. So this is what we mean by the neuron reaching its threshold in membrane potential. That's reaching the activation of sodium ion voltage gated channel. So here it is, step by step, what happens in an action potential. I'm repeating myself over and over again because students always get so confused about all this stuff, especially if these are your first exposure to this action potential stuff, okay? So at resting membrane potential of -70 millivolts, it is what we call resting membrane potential. The axon lemma contains your voltage-gated sodium channel and potassium ion channels. At this moment, it's resting. So both channels are closed, okay? So make sure you understand this pretty well because there will be a lot of questions on the exam saying, okay, the membrane is at resting membrane potential. Which channels are open below? Or this membrane is at +10, okay? So at that millivolts, what channels are open within the neuron, okay? So this is what you need to understand in terms of the action potential. All right, so let's say graded potential was large enough to get to -60 millivolts, which then trigger activation of the sodium ion channel and opens it up. Sodium ions then rushes into the neuron, and we know that it rushes in very rapidly because it's driven by that large electrochemical gradient we talked about earlier for sodium ions, and rapid depolarization happens. So that depolarization leads to the membrane potential gets more positive, more positive, more positive until it reaches its peak at positive 30 millivolts, okay? And once it reaches positive 30 millivolts, your sodium ion channels will close now, blocking any more sodium ions from moving in. And the other thing that happens is positive, your voltage-gated potassium ion channels now will open. Potassium ion then is able to flow out of the cell according to its electrochemical gradient we talked about with potassium ion, and it starts shifting the membrane potential back to resting membrane potential, because potassium is a positive charge. If you eject more positive charge out of the neuron, it's going to climb back down to be more negative. Now, what happens with potassium channel is that, once it only starts closing when the membrane reaches negative 70 millivolts. But, that's starting of closing of the potassium channels. So, it all doesn't snap shut all of a sudden. It kind of takes some time for all of the potassium channel to be completely closed, but it only is triggered when it reaches negative 70. So that's why the ejection of potassium ion kind of overshoots the membrane potential a little bit and results in a lower membrane potential than what is supposed to be at resting. So we call this period hyperpolarization, because we're waiting for all the potassium ions to close, the ion channels to close. As we're waiting, more potassium ions are being still, moved out of the cell. So that's why you end up with even a more negative membrane potential than you are at resting. But once all the potassium channels have closed, the natural permeability of the neuron to sodium ions and potassium ion should bring the neuron membrane potential back to negative 70 millivolts. And now you're back to resting membrane potential, and the action potential is now officially over. Now, this is that refractory period I was talking about during that positive 30 millivolts time, or even during negative 90. Okay, these are two different refractory period. When it's in this period, depolarization period, we call it absolute refractory period, and then when it is in this hyperpolarization, we call it relative refractory period. I'll explain why that is when I start drawing this in terms of a graph. It's easier to explain there. Okay. But refractory period corresponds with when the plasma membrane of the neuron no longer responds to any more stimuli coming through until that resting membrane potential is reestablished after the action potential. Okay? So now, let me draw this change in membrane potential during an action potential in a graphical format so it's easy to remember for exam purposes. Now, let me draw the graph. Okay, so we're going to start by drawing the y-axis and the x-axis. This is time as it elapses, and then this is millivolts of the membrane. Membrane potential. Okay, so usually at resting membrane potential, it's at negative 70 millivolts for a neuron. Okay, so it's flat for a moment until a stimulus arrives and it starts climbing up. Okay, so this is arrival of stimulus. Okay, and it reaches that threshold, which is negative 60 millivolts. And it starts opening. Right here is when it opens. Okay, so sodium ion channels open. Okay, I'm not going to put channel, but you know what I mean. And then it climbs all the way up very quickly and peak at about positive 30 millivolts. Okay, so this is the peak that it will reach at, and this whole step here is called depolarization. Then it climbs back. I don't like it when it's curved like that. Oh, I can't erase. That's straight. Oh, there you go. Okay. And then now it climbs back down, okay? Because once you're at this peak level, what happens here is that sodium channel will close, potassium channel will open, okay? Once potassium channels open, potassium ions will go out of cell. This is when sodium ion into cell. Okay, so when potassium ions, which is positive charge, move out of the cell, it brings the membrane potential back down and even past that resting membrane potential point, okay, to negative 90 millivolts, and we call this hyperpolarization due to waiting for potassium ions channels to all close. Okay, so then once all the potassium ion channels close, then it slowly climb back up to resting. Okay, this is negative 70 millivolts back to resting membrane potential. Okay, all right, so you have area of resting membrane potential. Okay, so this is resting right here. Okay, and then it has that arrival stimulus, allows it to reach threshold, goes through the process of depolarization, peaks, comes back down, and repolarization. But repolarization always overshoots, leads to negative 90 millivolts, which is called hyperpolarization. And once all the potassium channels open, it will naturally climb back up to negative 70, which is your original resting membrane potential. Okay, so now with the refractory period, we call this area when the membrane potential is climbing up due to the sodium ions. So this whole area here, okay, we call this the absolute refractory period, which means during this time, all the sodium channels are already open. We cannot open any more than they are already open. So there's absolutely no arrival of a second stimulus when you're in that absolute refractory period. When you're in this period, this whole period here, okay, then this is called relative refractory period. Because technically speaking, now your sodium channels have already closed, which means as your membrane potential is climbing downwards, a second stimulus can actually arrive and cause the sodium channels to open again. Okay, so let's say this climbs back down to negative 65 or something, negative 65. Okay, so technically speaking, if a stimulus comes through and bring it back up to negative 60, we can open the sodium channels again and get another stimulus going through. Or even at negative 90, we can still technically, if the stimulus is strong enough to provide that extra boost in membrane potential, we can still activate a second action potential within this area. So technically speaking, I would say the relative refractory period is more so we're talking about this area, okay? When we're talking about negative 65 okay? When it gets below the activation or threshold of your sodium ion channel, okay? So this graph kind of summarizes the whole process of action potential. What are the steps involved? What happens during each step at certain voltage? What channels are open? Which channels are closed? Which ion is moving where? And what does absolute refractory period mean? And what does relative refractory period means? Okay? All right. And to put it all in a nutshell and to see things moving in and out, the actual ions during the movement, this animation video will cover the whole process of an action potential so you can visually see what's happening. Let me remove this.
How does the pain you experience when you burn your hand result so quickly in an action by your muscles? Many animals respond to environmental stimuli using specialized cells called neurons. A stimulus is detected by sensory receptors, and the body responds through motor effectors. These cells working together allow you to respond very quickly to threats. When you touch something hot, heat receptors of a sensory neuron detect the stimuli and send the information of heat to an interneuron in your central nervous system. From there, a motor neuron sends a response from your central nervous system to the skeletal muscles in your arm, causing them to contract and pull your hand away. The fundamental process of neural transmission that underlies this action occurs in all neurons of the body. Neurons transmit this information through changes in the electrical potential of the membrane by the movement of ions across the membrane. An electrochemical gradient governs the movement of these ions, resulting in an electrical impulse. The resting membrane potential in a neuron, when the cell is not firing an impulse, is established by the unequal distribution of sodium ions outside of the cell and potassium ions inside the cell, making the outside of the cell more positively charged compared to the inside. The electrochemical gradient is established and maintained by an enzyme called sodium-potassium ATPase. When a neuron is stimulated, sodium ion channels open and sodium ions flow into the cell. This leads to a change in the electrical potential across the membrane called depolarization. The depolarizing electrical potential travels down the dendrites and over the cell body. Multiple electrical potentials will combine at the axon hillock in a process called summation. If the depolarization is large enough, an action potential is triggered. Action potentials are all or none electrical impulses that maintain their amplitude and strength down the length of the axon. The action potential travels down the axon when the depolarization of an area of membrane causes adjacent voltage-gated sodium ion channels to open. The influx of sodium ions results in membrane depolarization along the membrane. After a short delay, potassium ion channels open and potassium ions flow out, repolarizing the membrane. For the neuron to fire again, the resting membrane potential needs to be reestablished. Sodium-potassium ATPase is used to move sodium and potassium ions against their concentration gradients, reestablishing the resting membrane potential. As the action potential moves down the axon, ions are diffusing only a short distance, allowing the signal to move quickly. At the axon terminal, the electrical impulse passes to another cell at a cellular connection called a synapse. The space between the presynaptic neuron and a postsynaptic cell is called the synaptic cleft. The presynaptic neuron contains signal molecules called neurotransmitters that are packaged inside vesicles. When an action potential reaches the end of a neuron, neurotransmitters are released by exocytosis from the neuron into the synaptic cleft. Neurotransmitters bind to the adjacent cell at receptor sites attached to ion channels. The channels open, allowing the movement of ions into or out of the effector cell, which alters its membrane potential, thereby transmitting the signal from the neuron to the effector cell. Because nerve impulses move very rapidly down the axon of a neuron, and move from cell to cell across synapses, you react quickly to a stimulus, like burning your finger.
Okay, so it's almost an hour. I'm going to stop it at this part here so we can continue and review this one last time in part 3 for chapter 12 video.